cytology, which we are going to be looking at cytology 1. Straight away we are going to look at the outline of our lessons today. We are going to read it for the part 1 of cytology. We are going to look at the overview of the cellular basis of life. We are going to look at the plasma membrane as well as the cytoplasm. All right, now looking at the overview of the animal cell. If you look at this animal cell here, look at the animal cell. The animal cell, if you look at it, it has a membrane. If you look at it, the membrane surrounds the cell, and within the cell you have a nucleus which is at the central position. Look at the nucleus at the central position, and between the nucleus and the membrane you have what we describe as the cytoplasm, which contain as many organelles. We can identify the various organelles. We have a lysosome, Golgi apparatus, we have mitochondria, and so many other organelles which we can see from our diagram. So straight away we are going to look at the brief history of the cell. We will say that the English scientist Robert Hooke first observed plant cells with a crude microscope in the late 1600s. So you have to note all of this, especially this history for those of you as you'll be writing general knowledge. These are important aspects which you are expected to know. We will go straight away by looking at what a cell is. A cell is the basic structural and functional unit of all living organisms. If you look at the house is actually what it is because it is made because of smaller blocks joined together to make up a complete house. So a cell, the human body, is made up of smaller unit that combine, so many of it combine together to form what we have as a human body. So you see, the cell is a building block of an organism. The cell is a building block of an organism, and the activity of an organism actually depends on both the individual and the collective activities of its cells. So we see that the human body actually has about trillions of cells, which have about 200 different cell types, which they differ in their shape, size, and function. For example, you have fat cells. They are spherical in shape, and if you look at the red blood cells, they have a disc shape, and you have nerve cells, they are branching. And as you can see, like I said, we have different kind of cells in the body, and each of them are performing different functions. Now, cells are classified into two types. We have what we call the prokaryotic cells, and we have what we call the eukaryotic cells. Now, prokaryotic cells are those cells whose nucleus lies naked in the cytoplasm, while eukaryotic cells are those whose nucleus are enclosed by a membrane. So they have membrane bound nucleus. So that is what we mean by they are eukaryotic. The word "eu" actually means true, karyotic means nucleus, so they have a true nucleus. All right. So if you look at classification of organism, you see that the organisms like the protozoa, the fungi, plants, and animals, they are all regarded as eukaryotes, while the bacteria, they form what we call the prokaryotes, because they don't have a well enclosed, their nucleus is not enclosed by nuclei membrane. So we can see the differences that exist between prokaryotic cells and eukaryotic cells. For prokaryotic cells, you can see we have mesosomes for respiration, while for eukaryotic cells we have mitochondria for respiration. And if you look at prokaryotic cells, you see that they have very few, and most of them don't have membrane. They have very few organelles, and most of them don't have membranes. While if you look at eukaryotic cells, you see that many of them have a membrane bound organelles. Now for prokaryotic cells, they have the 70S ribosomes, while the eukaryotic cells, they have 80S ribosomes. All right, now for prokaryotic cells, we see that they have circular DNA that are found free in the cytoplasm, while eukaryotic cells, they have linear DNA with proteins and RNA to form the chromosomes. So you can see the differences that actually exist between prokaryotic cells and eukaryotic cells based on different criteria chosen. Now we are going to have an overview of the plasma membrane. Remember, we said a cell is separated from its environment by a membrane, and the animal cell actually has a membrane which we call it the plasma membrane, and this plasma membrane has its structure which we are expected to know a lot as far as its structure is concerned, because if you look at most of the questions that you find in your concours, you find a lot of questions coming on cytology, about 10 questions coming for cytology. So I will advise you students to actually take note, especially on the structure of the plasma membrane. And if you look at the structure of the plasma membrane, you are going to we are going to see that it has a certain if you look at the structure, this is the structure of the plasma membrane. Looking at it, it has a structure which we describe it as the lipid it has a lipid bilayer, and this lipid bilayer, as you can see, you have you have a type of you have the phospholipid which has a tail, it has a tails and it has a head, and the tails are said to be hydrophobic, while the heads are said to be polarized and they are hydrophilic. That means what I mean by hydrophilic is that they are water loving, while the tails are water hating. That means they hate water, so they are repelled by water. That is why if you look at the arrangement of the cell membrane, is arranged in such a way that if you look at the polar head, they are facing it towards the intracellular milieu, while you look at the other part is facing in the extracellular milieu. Remember, the intracellular milieu is an aqueous medium. That means it is made up of water. That is why you see the heads are orientated in that manner. But now if you look at the way they are orientated, if you look at the way they are orientated in the extracellular milieu, you find that the heads, if you look at the heads, you see that they are facing the extracellular milieu because they are water loving. Now look at the tails, they have this orientation that are found within the middle. That means because they are water hating, so they are being repelled by water. Now the cell membrane, which we call it the lipid bilayer, is not only made up of the phospholipid, but it is also made up of other components, like we have glycoproteins, we also have glycolipid, and we also have cholesterol. Now each of these they have their functions that they play within the cell. Now if you look at it, you also have what proteins that are found there. There are some proteins that you can see them just the level of they are just facing, they are not going through the membrane, they are just at the top. That means they are facing the extracellular milieu. Those proteins are what we describe as extrinsic proteins. But now if you look at the proteins that are facing the intracellular milieu, there are proteins that we describe as the intrinsic proteins. Now we can see some proteins like this one is actually crossing through the membrane. We describe it as a transmembrane proteins, and each of these proteins, like I said, they have their functions, each of these components. Now if you look at this, like for example, some proteins, they have carbohydrate attached to it, which they form glycoproteins. Now you also have some other you have some lipid to which carbohydrates are attached to them, which you describe them as glycolipid. You have, like I said, we have cholesterol. Okay, now we are going to look at the functions of each of these various proteins, and also the cholesterol, the various components that make up the plasma membrane. So like I said, we have two distinct populations of the membrane proteins. You have what we describe as the integral proteins, and you have what we call the peripheral proteins. The peripheral proteins are those that face either the extracellular milieu or those that face the intracellular milieu, but the integral proteins are those ones that cross through the membrane. So proteins make up about half of the plasma membrane. These are assertions that every student is expected to know. Half of the plasma membrane. Now responsible for most of the specialized they're actually responsible for most of the specialized membrane functions. We have integral proteins, which are firmly inserted into a lipid bilayer. Some protrude from one membrane face only, but most are transmembrane proteins that span the entire width of the membrane and protrude on both sides. Now if you look at these transmembrane proteins, they are mainly involved in transport. You see, they form what we call channels, other form what we call pores, through which water water soluble molecules or ions can move through. Remember, we said the inner part of the membrane, is made up of what? It is made up of the 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 tail, which is made up of a lipid, and that lipid tail is water heating. So there is no way by which water soluble substances can pass through the membrane. So the only way they can pass through is through these channels, through these protein channels, and through these pores, through these pores. So that's the only way they can pass through this membrane. And we have other, and some of these proteins, they, like I said, they act as channels which to allow these water soluble substances to pass through, and ions as well. Then you also have others acting as carriers. That means they bind to substance and then they are able to move the substance through the membrane. Now other acts as receptors for hormones or other chemical messengers, and they are able to relay messages to the cell interior. That process is what we describe as signal transduction. All right, now we are going to look at the peripheral proteins. We said they are not embedded in the lipid. They are attached loosely to integral proteins or membrane lipids, and they are easily removed without disrupting the membrane. And we said they act as enzymes. Some are involved in mechanical functions like changing the shape of the cell, others for muscle contractions, other helps in linking one cell to the other. All right, now we have the glycocalyx. The glycocalyx, we said it is actually a sticky carbohydrate-rich area at the cell surface, and it is actually enriched with glycolipids and also glycoproteins secreted by the cell. They provide highly specific biological markers. That means they are for recognition. That means, for example, if you take for example the sperm cell, for the sperm cell to be able to recognize the egg cell to doing fertilization, there is a layer which we call the glycocalyx layer on the egg cell that enables them to be able to identify the egg cell. So the for identity. Okay, we also have cells of the immune system. They are also possess glycocalyx. So we are going to look at the various specializations of the plasma membrane. We are going to look at microvilli, which are small hair-like structures, they are finger-like structures which we found at the level of the plasma membrane. They project from a free or exposed cell surface. So they actually increase the cell surface membrane. Membrane area. That means when as a result of increase in cell surface membrane area, they help in absorption. Cells that possess microvilli, like the cells found at the level of the ileum, they are involved in absorption. These are cells that are involved in absorption. Now we are going to talk about cell junctions, but in this cytology we are going to see cell junctions under histology, which will be coming up subsequently. We are going to look at membrane transport, the various processes by which substances move in and out of the cell. Substances actually move in and out of the cell by two processes. You have the passive process that doesn't require energy, and you have the active process that requires energy. Now we are going to look at the various passive processes, and we look at the various active processes. Now under passive processes, we have two main processes. We have diffusion, and we have filtration. Diffusion, will define it as we are going to define diffusion as the process by which substances move from a region where they are highly concentrated to area where they are less concentrated down a concentration gradient. Now diffusion is divided into different types. You have simple diffusion, which involve non-polar and lipid soluble substances that diffuse from the region where they are highly concentrated to the region where they are less concentrated. We have oxygen, carbon dioxide, fat soluble, vitamins, and so on and so forth. Now we have facilitated diffusion, which is diffusion that is aided by molecules. Notably, we have glucose and other sugars, amino acids, and ions, which are transported passively even though they are unable to pass through the lipid bilayer. So instead they move through the membrane by passive active transport process, which we call it facilitated diffusion. So we are going to look at carriers. We have a carrier is a transmembrane integral protein that shows some specificity for molecules of a certain polar substance or class of substance that are too large to pass through the membrane channels, such as sugars and amino acids. So we have channels that are open. Also, they are also enables for movement, for transport of transport substances such as ions, water, and aqueous substances that are soluble in water. That's what I mean. So we have different kind of channels. We have leakage channels, and we have gated and uh, control channels. Leakage channels are those that are always open and simply allow ion or water fluxes according to their constitution gradient, while you have others like the gated channels, which are channels that by which are channels that they are closed, and so they can only open as a result of certain signals. Now we are going to look at another type of diffusion, which we say osmosis. It's a special kind of diffusion which involves the movement of only water molecules from a region where they are highly concentrated to a region where they are less concentrated down a concentration gradient through a selectively permeable membrane. That means a membrane that is selective to allow only water molecules to pass through it. So water also move freely and reversibly through water specific channels constructed by transmembrane protein called the aquaporins. Aquaporins, we can find them at the level of the kidneys, even in red blood cells. They are abundant in red blood cells. So we are going to look at some important terminology such as osmolarity. So every student should be able to differentiate what osmolarity. As an assignment, you have to look for the difference that exists between osmolarity and osmolality. Now we are going to look at what we mean by isotonic solutions, hypertonic solution, and hypotonic solution. Isotonic solutions are solutions that have same concentration, while hypertonic solution is a solution that has a concentration that is greater than the concentration of another solution. So comparing two solutions, one with a higher concentration than the other, with respect to the other, we call it is that a solution. If we have, for example, two solutions, solution A, we have solution B. Solution A, if solution A has a concentration that is higher than the concentration of solution B, we say solution A is hypertonic to solution B. Now on top of hypotonic, the reverse is true. Alright, under active processes, we are going to look at active transport, vesicular transport. Active transport, we are going to say that active transport involves the movement of molecules from a region where they are less concentrated to a region where they are highly concentrated with the use of energy which comes from ATP, from the hydrolysis of ATP. But now we have facilitated we have two kind of active transport which you are going to see. We are going to see primary active transport, and we have secondary active transport. Primary active transport requires energy that comes directly from the hydrolysis of ATP. For example, we have the sodium potassium pump is a perfect example of a primary active transport. So now we'll talk of secondary active transport. It is a transport that is driven indirectly by energy stored in ionic gradient created by operation of primary active transport pumps. So that means in this case we have a scenario where energy is stored within a gradient and is used to transport a substance. So the energy does not come directly from the hydrolysis of ATP. So now we are going to see under a secondary active transport, we have the sympod system, which involves the transport of two substances in the same direction. Now we have the antipod system, which involves the transport of two substances across the membrane in opposite directions. So now we are going to look at the cytoplasm, look at the various organelles that are found within the cytoplasm. Like I said, the cytoplasm forms the house of organelles. That's where the organelles are living. So it is a cellular material between the plasma membrane and the nucleus. It is a site where most cellular activity actually take place. And if you look at the cytoplasm, it is made of a liquid part which describe it as a cytosol. And within the cytosol, we have water, and we have dissolved substance, we have proteins, we have sugars, salts, and other solids that are dissolved within it. Now we have the other known membranous organelles, that means organelles that lack membranes. Example, we have the cytoskeleton, the centrioles, and the ribosomes. They don't have membranes. I repeat, cytoskeleton, centrioles, and ribosomes don't have a membrane. But now if you look at other organelles like the mitochondria and the mitochondria, they have membrane, and some of them have double membrane. Yes, they have double membrane. All right, now we are going to look at the mitochondria. The mitochondria, a thread-like or shaped membranous organs is found in living cells, and it is referred to as the house of the cell, because that is where ATP is being produced during respiration. And busy cells like kidneys and liver cells will have hundreds and hundreds of mitochondria, whereas inactive cells don't have that, like lymphocytes. They don't have mitochondria, except so many mitochondria. So if you look at the structure of the mitochondria actually is a double membrane organ. And if you look at the mitochondria, it is also described as a semi-autonomous organ. Why is it referred to as a semi-autonomous organ? Because it has its own DNA, so it's able to replicate on its own. So if you look at the mitochondria, like I said, it has two layers. You have this part, this external part, and you have this internal part. The internal part, the reaction that you have, what we call the internal part. They have this the internal part, this part we call it the matrix, and here we have small this foldings which we call a cristae. And now within you need to know where glycolysis actually take place and where electron transport chain takes place, where the Krebs cycle actually also take place. So you have to know where glycolysis takes place, the Krebs cycle takes place, and electron transport chain. As an exercise, all my students, you have to do research on that. Not everything must be told to you. Every student must learn to do research. We are not training you for you to go to be a weakling, but you need to be someone that is strong, be able to do a lot of research. All right. The mitochondria, we say, is enclosed by two membranes. We have the outer membrane and you have the inner membrane. We say they contain their own DNA. We have already discussed on that. We are going to look at ribosome. We say they are small dark staining granules that are composed of proteins and a variety of RNA called ribosomal RNA. So we have, each ribosome has two globular subunits that fit together like the body and the cap of an acorn. So ribosome, actually they are site where protein synthesis actually takes place. So we are going to look at the rough endoplasmic reticulum. We have the rough endoplasmic reticulum and we have the smooth endoplasmic reticulum. So those are the two different endoplasmic reticulum that we have. Now the rough endoplasmic reticulum has its own function. And on the rough endoplasmic reticulum, like I said, it is where protein synthesis really take place. But now for the smooth endoplasmic reticulum, is in communication with the rough endoplasmic reticulum and consists of what? Tubules arranged in a looping network. Its enzymes that is play no role in protein synthesis. Instead, they catalyze reaction involving the following processes. They are not involved. So the smooth endoplasmic reticulum, they are not involved in what? Protein synthesis, like the rough endoplasmic reticulum. Now the smooth endoplasmic reticulum, that involved in lipid metabolism, cholesterol synthesis, and synthesis of lipid components of the lipoproteins. So synthesis of steroid-based hormones such as sex hormones, such as testosterone, synthesizing cells of the testes are full of smooth endoplasmic reticulum. You'll find a question in the conclude will ask you which of the a cell which leads to the which produces steroid hormones possess majority of the following. A, they'll put rough endoplasmic reticulum. B, nucleus. C, this. So you should know that it should be the smooth endoplasmic reticulum, because all those what produces lipids, which that does lipid metabolism, is the smooth endoplasmic reticulum. Now it also functions in detoxification of drugs, some pesticides, and even some carcinogens at the level of the kidneys and even in the liver. Now the Golgi body, which consists of stacked and flattened membranous sacs, and if you look at it, they are actually they help in the modification of proteins, and so that's their function. So they help in modification, packaging enzymes for lysosomes, sorting all kinds of material to deliver it to the plasma membrane. So those are their functions of the Golgi body or the Golgi apparatus. So we are going to look at lysosomes. We say they are spherical membranous organelles that contain digestive enzymes, and we find the majority in phagocytic cells, where the enzyme help to digest bacterial cells and cell debris. So they work best in acidic conditions, and thus they are said to be acid hydrolysis. And like I said, they function in what? Digestion, removal of damaged organelles, and the cell self-destruction. They can cause suicide. they destroy the cell itself. So that's what we call cell suicide. So the organisms are responsible for cell suicide are lysosomes. So you have peroxisomes that contains oxidases and catalases. They contain those enzymes, and remember, catalase help to hydrolyze hydrogen peroxide producing oxygen. The cytoskeleton, they are an elaborate series of rods running through the cytosol. Their network is actually made up of bones, muscles, and ligaments by supporting uh, structures cellular structures that provide machinery to generate various cell movements. We have three principal types of cytoskeleton. You have the microtubules, the microfilaments, and the intermediate filaments. So as an assignment, I want you to describe the different portions of the cytoskeleton, describe the cellular extensions and distinguish between them, giving examples of their locations and functions in the human body. So every student has to do that. So we are going to, in our next edition, we are going to look at Cytology part 2. Thank you for your kind attention. This was Cytology 1, presented to you by Sir Alain Dong, student medical doctor from the Faculty of Medicine and Biomedical Sciences of the University of Yaoundé 1. Thank you.